Hello everyone. So this is the 11th problem solving session for the NPTEL course Experimental Biotechnology. The course instructor is Professor Vishal Trivedi from IIT Guwahati and I am Shrija Ghosh, a PMRF from IIT Kanpur. So today's topic is all about DNA sequencing, protein sequencing, and then we'll move on to two new techniques, northern blotting and southern blotting. So till now we have discussed about electrophoresis techniques and we had discussed about western blotting, which was used to detect certain proteins. Okay, so similar to that, there are other techniques that are used to identify RNA or DNA sequences, which are basically northern blotting and southern blotting. Okay, and then we also have completed polymerase chain reaction in the previous session, where we discussed how to amplify a particular sequence of DNA. Now, during that amplification process, we saw that there could be chances of error happening in the sequence. So, let's say I want to know where the error is happening. So how am I going to do? I need to develop a platform where I can sequence my DNA, my DNA product that I am getting after amplification. Or let's say I have identified some gene, like I have isolated some gene. So now I want to know the sequence of that gene. So for those purposes, we need to know how to determine the sequence of the DNA. So DNA is made of 4 different bases: adenine, thymine, guanine, and cytosine, A, T, G, and C. So how do we decide? Like how do we determine what is the exact sequence of this DNA? So DNA is a polymer of these 4 different nucleotides, right? So we need to determine how to determine the whole sequence of a DNA. Similarly, like we have DNA, we have proteins also. The another biomolecule that we are studying is protein. So proteins also are polymers of different amino acids. So now, like in case of nucleotides, we had 4 different kinds of nucleotide: adenine, thymine, guanine, and cytosine. In case of amino acids, we have 20 different kinds of amino acids. So now the complexity now increases as where in case of DNA we had only 4 bases to detect. In case of protein we have 20 amino acids to detect. So but sequencing, like the idea behind sequencing remains same, the purpose behind sequencing remains same. We want to know the sequence of a particular protein, a particular DNA, so we'll apply such techniques. So we'll go through different questions related to sequencing and related to northern and southern blotting, and along with those questions I'll keep on explaining the techniques as well. So I hope this session would be helpful to all of you. Let's begin with the questions. So starting with the first question, what is the purpose of dideoxynucleotides in Sanger sequencing? What is the purpose of dideoxynucleotides in Sanger sequencing? Option A, to elongate the DNA strand during synthesis. Option B, to initiate the DNA synthesis. Option C, to terminate DNA synthesis at specific position. And option D, to label the DNA fragments for detection. What is the purpose of dideoxynucleotides in Sanger sequencing? So first thing is, what are dideoxynucleotides? So if you remember, DNA is basically ribonucleic acid, right? The full form of DNA is deoxyribonucleic acid. And for RNA we only have ribonucleic acid, right? So this is the RNA, and when we add deoxy part, this is DNA. Why do we write deoxy? So basically DNA, as I mentioned, it is made of 4 different types of nucleotides. Nucleotides have a sugar backbone, a phosphate group, and a nitrogenous base attached to it. So in that sugar backbone that we have, so this will be the first carbon, this is the second carbon, this is the third carbon, this is the fourth carbon, and then we have another carbon to which the phosphate group is attached, right? So this is a 5 carbon sugar, ribose sugar, that is the backbone of a nucleotide. Okay, now this second and third carbon has OH groups in case of RNA. Okay, in case of DNA, as we can see in the name, we have deoxy. That means one of the oxygen is removed. So in case of DNA, the sugar base, the second carbon of the sugar base does not have the oxygen. So this is kind of the structure that we have in case of DNA. Okay, so that's why it is named as deoxyribonucleic acid or deoxynucleotide. This is a deoxynucleotide, that is, so from this first carbon we will have the nitrogenous base attached. So this is DNTP. Now in this question, if you see, we have DDNTP. What do we mean by dideoxy? That means 2 of the oxygens are removed. That means along with the second oxygen, the third oxygen is also removed from this hydroxyl group. So now we will have the sugar base with both the carbons having only hydrogen instead of OH. Okay, so we will have the base here and we will have the 5th carbon here. Okay, and now this third oxygen group, the hydroxyl group, is very important when we have to make a complete DNA sequence. So DNA, if you remember, we write the DNA as 5' to 3'. Okay, why do we do so? Because this is the third carbon. The hydroxyl group of the third carbon is actually attached to the phosphate group of the 5th carbon. So the DNA kind of starts with the 5th carbon and ends at a third carbon, like ends at this hydroxyl group of the third carbon. So we can have the phosphate group attached to the 5th carbon, then we will have the whole base, and from this third carbon, the OH, this OH will now go and make a phosphate bond with the 5th carbon of the next base. Okay, and that's how we have the DNA sequence. So if I remove this oxygen from the third carbon and make it dideoxynucleotide, what will happen? The DNA synthesis will be terminated, right? So the correct answer is option C. Why the DNA sequence will be terminated? DNA synthesis will be terminated because now there won't be any hydroxyl group to make the phosphate bond with the next nitrogenous base. So when the DNA synthesis is going on, like in a PCR reaction, if you remember, we had a DNA template and then the primer, and then one by one each DNTP was incorporated according to the complementary to the sequence of the template DNA, right? Now this incorporation will get halted if we add DDNTP. So let's say the 4th base that is added is a DDNTP. So what will happen after that? The 5th base cannot be added because there won't be any hydroxyl group present to make the phosphate bond, phosphodiester bond with the next incorporating nucleotide triphosphate. Okay, so that is why third option is correct. The purpose of dideoxynucleotide is to terminate the DNA synthesis at specific position. How the specific position is how how do we get that termination at a specific position? I'll show. So this is what I was talking about. So this is the ribose sugar backbone. We have the nitrogenous base, we have the fifth carbon that has the triphosphate group, so that will make the phosphodiester bond. Now this particular OH is important in case of making the bond with the next nitrogenous base. So if you see here, so this is one DNTP, okay, this is the phosphate group, triphosphate group, fifth carbon, and we have the whole nitrogenous base, and this is the nitrogenous base, and we have the whole ribose sugar. And then from the third carbon, the OH of the third carbon will make the phosphodiester linkage. This is the phosphodiester linkage with the phosphate of the fifth carbon of the next nucleotide. Okay, now in this way the reaction goes on. Now once we remove the oxygen of one of the nucleotide, that means we make it DDNTP, what will happen? The next base cannot enter the chain and the synthesis will be terminated at this point. Now what do we do with this information? So what do we do when we set up a Sanger sequencing reaction? What do? We have a template DNA. So here I have the figure. So let's say this is my template DNA, the blue one, and this is, I have added a radioactively labeled primer. And now normal PCR reaction, how we were doing, we were adding the DNTPs, we were adding the polymerase, and then we were doing a thermocyclic reaction. Similarly, we will do the same thing here. We will add the DNTPs, we will add the DNA polymerase and let the reaction begin. But along with all these 4 DNTPs like DATP, DCTP, DTTP and DGTP, along with that, so we'll make this master mix in one tube and divide it in 4 vials. Okay, so all 4 vials will have all 4 DNTPs. Along with these 4 DNTPs, each vial will get one of the dideoxy nucleotide. That means one of them will get DDATP, one of them will get DDCTP, one will get DDTTP and one will get DDGTP. Okay, then the one in which I have added DDATP, what will happen? So in this sequence that you see here, the primer has bound this part, and then now incorporation of the nucleotides will start. So in front of G we will have an incorporation of C, in front of C we will have incorporation of G, in front of T we will have incorporation of A. Now since this mixture has both DATP and DDATP, what will happen in this vial? Either DATP can get incorporated, so adenine will get incorporated. In that case, the reaction will proceed. In case DDATP gets incorporated, that is the dideoxy adenine triphosphate gets incorporated, what will happen in that case? This adenine will not have the OH group, and because of that the chain will terminate and we will get a short fragment, that is that the sequence of which will be T, T, G, T, C, G and A. That's it. Okay, the sequence will get terminated at that point. So here you can see this is the fragment. But if the reaction proceeds, then we will get, and then next is also A. So if in that case DDA gets incorporated, the chain will terminate there. In the third case, the chain can get terminated at this position because here we have T, so the complementary will be A. So this kind of reaction can go on in this first vial. Similarly, in case of second vial, wherever C is getting incorporated, the chain can get terminated there, or chain can proceed if the DCTP gets incorporated, and it will get terminated at another position where C has to be incorporated. Once all the 4 fragments are, once all the 4 reactions are completed, what do we do? So what do we do is we separate the fragments, all these fragments on agarose gel electrophoresis. So, if you remember from the electrophoresis chapter, electrophoresis session, we had discussed how agarose gel electrophoresis can be used to separate DNA molecules based on their different molecular sizes. So, smaller the size, faster it will run. The longer the size, the slower it will run on the gel. So, if the first strand that is there, first base that is there, that has to be incorporated here. So, the first base that has to be incorporated is a C. In this case only, if DDCTP gets incorporated in this vial, in the second vial, we will get the smallest fragment at the very base. Okay, then the second largest fragment, then the third largest fragment, and so on. So, when we go from bottom to up on this agarose gel, we can actually deduce the sequence of the DNA. So, if you see here, so DDCTP, the smallest fragment is from DDCTP. That means after the primer, the first base is C. Then the second fragment is in the DDGTP. That means the second base that is there is G. Similarly, the third one is A. Then again, fourth one is A. Fifth is G. Sixth is T. Seventh is C. Eighth is A, and ninth is G. So, this is the sequence of the complementary strand of the template that we need to sequence. So, what we do, we simply take a complement of this particular strand, and that will be the sequence of our template, the sequence of the DNA that we wanted to know. Okay. So, this is how Sanger sequencing proceeds. So, you have mixture of DNTPs, and in each vial one of the additional dideoxy nucleotide phosphate is added in order to see where the chain termination is occurring, and based on the different sizes of the DNA fragments, we can deduce the structure, deduce the sequence of the DNA. Okay. And since we have used here a radioactively labeled primer, we can visualize this gel by using autoradiography. So, basically we can see under X-ray, we can see the radioactivity and the bands, and then visualize the gel based on autoradiography. Okay. So, I hope this is clear to everyone. I'll move on to the next question now. Question number 2. Strand separation for DNA sequencing is done to dash. Option A, to sort the fragments according to their size. Option B, to sort the fragments according to their molecular weight. Option C, to obtain the consecutive sequence as per termination. And option D, to obtain specific lanes of sequencing. Strand separation for DNA sequencing is done to sort the fragments according to their size. Option B, to sort the fragments according to their molecular weight. Option C, to obtain consecutive sequence as per termination. And option D, to obtain specific lanes of sequencing. So, the correct answer here would be to sort the to obtain consecutive sequence as per termination. So, as I've already explained in the Sanger sequencing, what we are doing, we are basically separating the strands based on their sizes to get the sequence. So, the smallest band will be the first base that will be incorporated, then the next, and like that we go higher in size, right? So we are doing the strand separation by doing agarose gel electrophoresis, where we are separating the fragments based on their sizes, but we are doing so to obtain the consecutive sequence as per the termination. So wherever the termination will happen, that would be the fragment size, and based on the size the separation will occur, and we can actually deduce the sequence. Okay, so nowadays, instead of running 4 wells of gel separately, so in the previous diagram you saw we had a agarose gel and we had 4 wells, 1, 2, 3 and 4. Each well had 1 reaction. So first well had DD-ATP, then the second well had DD-CTP, then the third well had the TTP, and then fourth well had the DD-GTP. Okay, so that is how we did. But nowadays we generally use capillary gel electrophoresis, where each of these DD-NTPs are basically fluorescently labeled. Okay, so wherever the termination is happening, so if the termination is happening at A, that will give one particular color. So A here has green color, then C has blue color, G has yellow color, and T has red color. So all these have different colored fluorescence, and based on the fluorescence that is detected, we can actually see what is the sequence of our DNA. So if this is the template DNA, this is the original DNA. Now when the sequencing will start, each and every fragment will each and every nucleotide, wherever the termination is happening, will have a particular fluorescence. Okay, so wherever this DD-ATP will be incorporated, will give a green signal. Wherever DD-TTP will be incorporated, it will give a red signal, and so on. So in one single well we can run this sample, and when each sample reaches the detector part, the signal is recorded as a chromatogram. So you can see in this graph, this is called a chromatogram because it's a colored photograph. Okay, that we are going to analyze. So you can easily see that, okay, this is a yellow peak, that means it's a guanine. It's a green peak after that, that means it's a adenine. Then we have C, then T, then again G, A, G, C, T, G, T and T. So you can see this becomes easier than running a electrophoresis gel with 4 wells, and then after running you visualize the gel and then analyze the sequence manually. So this will be more of a automated system. So this is the newer technology that is generally done for sequencing any kind of DNA. So let's say I am doing cloning. Okay, so I've cloned a particular gene in a pet vector system. So in a vector I have cloned my particular gene sequence, and now I want to see if my cloning is done correctly or not, if I have successfully incorporated the gene in the vector. So what will I do? I will give this whole plasmid for sequencing, for Sanger sequencing, and they will use primer somewhere here. The primer will anneal and it will keep on incorporating each bases, and wherever DD NTPs are incorporated of that particular color, it will give a chromatogram, and I'll give a whole chromatogram like that, and I can actually see the sequence from this part to all the way wherever the reverse primer will bind. So reverse primer, let's say, has bound here. So all the way from here to here, I can actually see the sequence of my DNA of interest. Okay, so this is all about Sanger sequencing. Moving on to the next question, question number 3. What is the typical order of steps in the Maxim Gilbert sequencing method. Option A, chemical modification, gel electrophoresis, DNA cleavage, visualization. Option B, DNA cleavage, chemical modification, gel electrophoresis, and then visualization. Option C, gel electrophoresis, chemical modification, DNA cleavage, and then visualization. Option D, visualization, chemical modification, DNA cleavage, and then gel electrophoresis. So what is the typical order of steps in the Maxim-Gilbert sequencing method? So do we do chemical modification first, or DNA cleavage first, or gel electrophoresis first, or visualization first? So what do we do first and what do we do last? So the first step logically is DNA cleavage, then comes the chemical modification, then comes the gel electrophoresis where you separate different bands, and then comes the visualization by when you will visualize the gel that has separated different fragments, and then you can do the analysis of the sequence. So this is the typical order of steps. So first, so why do we do DNA cleavage? Basically we do DNA cleavage to make longer DNA into shorter fragments so that we can get a proper sequence and there will be lesser error. Okay, so if we have a very long DNA sequence, and if we do like even if we do a Sanger sequencing kind of technique, what will happen, like it has certain limit, like after certain point it will not be able to sequence accurately, like it will not be able to incorporate the nucleotides accurately. Similarly, in case of Maxim-Gilbert sequencing method also, since it is based on chemical modification, if you take a very long DNA, the modifications will not be happening very uniform in the whole DNA. So it's better to cleave it into small pieces. It's like breaking down one long sentence into 2 or 3 short sentences so that it is easy to understand. Okay, if I write one sentence, one very long sentence, it would be very difficult for the audience to understand what I want to convey. Okay, so it's better to break that sentence into 2 or 3 short sentences so that it is clear to everybody, like what I want to say or what I want to convey. Similarly, for a very long DNA sequence, it's better to cleave it off into small DNA fragments, which can be then used for sequencing. So you sequence each fragment individually, and then since the fragmentation is random, you can actually do overlapping and see. So let's say this is one long DNA that needs to be sequenced. So what I do, I will make random fragments. So this could be one fragment, then I'll just change the color. So this could be one fragment, then this could be the second fragment, the ones that are bracketed by green, then this could be the third fragment, and this could be the fourth fragment. Okay, so based on the overlapping sequences, now we can actually determine what is the DNA sequence of the whole. So you can see here you will have one fragment like this, then you will have the second red fragment like this, then the green fragment will be like this, the blue fragment will be like this, then the purple fragment will be somewhere between the red and the green one, so something like this. And by seeing all these overlapping sequences, you can actually align the DNA and you can determine the sequence of the long DNA easily. So that's why we do DNA cleavage. Then comes chemical modification. Why are we doing chemical modification? So in Maxim Gilbert sequencing method, we do 4 types of chemical modification to determine whether it is a adenine, guanine, cytosine or thymine. Okay, so that is the principle behind Maxim Gilbert sequencing method. So in case of Sanger sequencing, we were adding DDNTPs and the chain termination was happening. In this case, the chain will be broken at the modified nucleotide. Okay. Then comes the gel electrophoresis. Once you have done the modification and you have cleaved at the modified sites, you will get small fragments, and then you can separate those fragments on the gel electrophoresis. And then how we were reading the sequence from a like gel that has been run after Sanger sequencing. Similar to that, we can analyze the gel of a Maxim Gilbert sequencing. Okay, so these are the 4 types of reactions that are done to identify the different different bases in the DNA. So the first reaction is with dimethyl sulfate. What it does, it modifies the guanine residues. Okay, so wherever G will be there, dimethyl sulfate will modify it. Then the second reaction is with formic acid. So wherever formic acid, what will formic acid do? Formic acid will basically act on the purine nucleotide. So what are purines? Guanine and adenine are the purines. Thymine and cytosine are the pyrimidines. Okay. So formic acid will act on the purine nucleotide. So it will modify both G and A. So if I have a sequence A, T, G, C, G, G, T, whatever sequence we have, okay, just a random sequence I wrote. So wherever adenine will be there, wherever adenine will be there or guanine will be there, those places will get modified when treated with formic acid. Okay, when treated with dimethyl sulfate, only guanine will be modified, not the adenine. Then third reaction is with hydrazine. So if we treat this DNA with hydrazine, it will break the pyrimidine ring. So that means thymine and cytosine, wherever T and C will be there, will get modified. And the fourth reaction is the same reaction, hydrazine reaction, but in presence of salt. So in presence of salt, only cytosine ring is broken. In absence of salt, both thymine and cytosine rings get broken. So both thymine and cytosine gets modified, but in presence of NaCl, only cytosine gets modified. So we have 4 types of reaction here. We have guanine, we have G plus A, we have T plus C, and we have the C reaction. After these base modification, piperidine is used to basically catalyze the cleavage of the phosphodiester bond next to the modified nucleotide. So let's say this sequence, this particular G is modified. That means the piperidine will do the cleavage after this G. Next, this particular G is modified, so the cleavage will happen after this. So now you can see, like it's Sanger sequencing, here also we are getting different size fragments, and based on these different sizes, it can be separated on the gel. So the smallest fragment will run fastest and the largest fragment will run the slowest. And then you can see, once you have all the sequences sorted, you can actually analyze the sequence of the complete DNA. So here you can see the A reaction, C reaction, G reaction and T reaction. So A reaction is basically A plus G, where the purine were getting reacted by formic acid. Okay. T reaction is basically T plus C. So C reaction is the one with NaCl and this is the one without NaCl. So without NaCl, both thymine and cytosine gets modified. With NaCl, only C will get modified. So you can see the modification is happening at different places. So the chain got terminated here. So we have different size of fragments, and then we can like we did in Sanger sequencing, we can have 4 wells for 4 different reactions, and then once we have the different fragments, now we can analyze the gel. So here you can see that smallest band is there in the C reaction and T plus C reaction. So both of them have cytosine in common. So the first base that is there, since it is there in the C reaction alone also, that means the band that we are seeing in the T plus C reaction is actually the band due to the nucleotide sequence C. Next is present in G reaction and A plus G reaction. That means the common is G, so G will be the next nucleotide base. Then after that we have a band at T plus C reaction, but that same band is not there in C reaction. That means this band is coming due to T, presence of T. Similarly, if you go like this, you can determine the complete sequence of the DNA. Okay, so both Maxim and Gilbert and Sanger sequencing, the end analysis is same. In Sanger sequencing, we had 4 wells for ATGC. In Maxim and Gilbert, it is slightly more complicated, where we have 4 wells with one having A reaction that also shows the background reaction for G. So we have to compare it with the G reaction, and the T and C reaction has to be compared with the C reaction alone. Okay. So I hope this concept is clear to everyone. I'll move on to the next question. Question number 4. What is the primary purpose of using dithiotritol in protein sequencing? Option A, to determine protein secondary structure. Option B, to cleave peptide bonds between amino acids. Option C, to reduce disulfide proteins in the to reduce disulfide bonds in the protein. Option D, to determine the protein's molecular weight. What is the primary purpose of using dithiotritol in protein sequencing? Option A, to determine the protein secondary structure. Option B, to cleave peptide bonds between amino acids. Option C, to reduce disulfide bonds in the protein. And option D, to determine the protein's molecular weight. So the correct answer is to reduce disulfide bonds in the protein. So dithiotritol is used to reduce the disulfide bonds. So this you have, if you remember from the electrophoresis session also, we had discussed in SDS page that we use DTT or beta-mercaptoethanol to reduce the disulfide bonds. So here also we are using it for the same purpose. We are using it for the reduction of the disulfide bonds on the protein. what DTT does is, so this is a example where the cysteine residues have formed disulfide bond. So if you remember, cysteine, the side chain of cysteine has a SH group. Okay, so this SH group, two cysteine residues can form a disulfide bond where these two SH groups can come close and make a disulfide bond, that is S bonded to another S, sulfur bonded to another sulfur. Okay, now this disulfide bond can be broken by using dithiotritol. So it can actually reduce this disulfide bond and two cysteine residues will be having free SH groups. So this is just one of the steps that is done before doing the protein sequencing. So why do we do this? Basically, if we have a protein 3D structure of the protein, so you know protein is a sequence of amino acid. Okay, it's a polymer of amino acids joined together like this. Okay, now this is the second, you can say this is the primary structure of the protein. Then this protein gets folded into secondary structure and then tertiary structure. So finally we have something very complex, which is a completely woven kind of structure. So if we want to sequence this, we first need to entangle this whole thing into a straight primary structure of the protein. Then only we can start reading from one end, and we can start reading the amino acids from one end, and we can write the sequence of the protein, right? So for that, first we have to entangle this whole thing into one single string. So to entangle this whole thing into one single string, we have to break the linkages that are there, that are holding this structure up. So one of such linkages, disulfide linkages. So we need to break this disulfide linkage so that we can entangle this whole 3D structure into a straight single string, so that we can read the amino acid sequence from one end. Okay, another reason is, so along with the entangling the whole 3D structure, even in the primary structure we have, like if we have one cysteine here, okay, and then we have another cysteine here after 4 or 5 residues, these two cysteines can make us disulfide bond. So what will happen if we don't break this disulfide bond? When I am reading, I might miss out this part of the protein. So I might not read the amino acids present between these two cysteines, because this part will not be separated completely. So basically, to separate the whole protein chain into one single strand, so that we can read the amino acid sequence of that protein, we need to break the disulfide bond, and that is generally broken by using dithiotritol or beta-mercaptadinol. You can use it to reduce the disulfide bonds. Okay, so once these disulfide bonds are broken, they are actually protected by carboxymethylation or by iodoacetate. We do this protection so that they do not join back again. So once I entangle this whole 3D structure into a single string, again these two cysteines can form the disulfide bond once the conditions are appropriate. So to prevent this formation of disulfide bond again, because we need to read the sequence, so we cannot afford to have a disulfide bond anywhere. So for that, we break the disulfide bond and immediately we protect these sulfur groups. How do we protect? We basically add a carboxymethyl group to this sulfur, so that this sulfur cannot form another bond with another sulfur group, free sulfur group. Okay. Moving on to the next question, question number 5. Cyanogen bromide cleaves the protein sequence. Option A, after lysine arginine. Option B, after methionine. Option C, after leucine, phenylalanine, tryptophan and tyrosine. And option D, phenylalanine, tryptophan, tyrosine. Cyanogen bromide cleaves the protein sequence. Option A, after lysine and arginine. Option B, after methionine. Option C, after leucine, phenylalanine, tryptophan and tyrosine. And option D, after phenylalanine, tryptophan and tyrosine. So cyanogen bromide is an enzyme, is a chemical that can actually cleave the protein sequence after methionine residues. Okay, so if I have a long protein sequence, so wherever we will have the methionine residue, it can cleave after the methionine residue. So in case of DNA we were having different fragments, similarly in case of protein also, once we have a long protein sequence, we can have different fragments based on wherever cyanogen bromide is cleaving the protein sequence, and then these fragments can be sequenced separately, and later we can assemble the whole protein into one single unit. Okay, we can assemble the sequence of each peptide that is sequenced into one single protein. So basically in protein sequencing, first we break the disulfide bond, as I explained, to get a single string of protein, and then that will be cleaved into multiple peptides, small peptides. So this cleavage is done using different enzymes. So these are some of the reagents, some of the enzymes and chemical reagents. So we have trypsin that cleaves after lysine or arginine residue. So wherever in the protein sequence a lysine will be there or an arginine will be there, so you can see it is cleaving after lysine, arginine. Then chymotrypsin can cleave after the aromatic residues, so phenylalanine, tryptophan and tyrosine. So it can cleave after either a phenylalanine or a tryptophan or a tyrosine. Then pepsin can cleave after leucine, phenylalanine, tryptophan and tyrosine. Okay, and then cyanogen bromide, as I already explained, it can cleave after methionine. And then once we have these small peptides, so we have T1, T2, T3, T4, right? So these small peptides now can be used for doing the sequencing, and then later we can again assemble this whole thing into one string, and then we can determine the sequence of the whole protein. Moving to the next question, which of the following is a Sanger's reagent? Option A, 1-fluoro-2,4-dinitrobenzene. Option B, 3-fluoro-2,4-dinitrobenzene. Option C, 2,5-difluoro-2,4-dinitrobenzene. Option D, 2-fluoro-2,4-dinitrobenzene. Which of the following is Sanger's reagent? 1-fluoro-2,4-dinitrobenzene, 3-fluoro-2,4-dinitrobenzene, 2,5-difluoro-2,4-dinitrobenzene, or 2-fluoro-2,4-dinitrobenzene. So the correct answer is option A, 1-fluoro-2,4-dinitrobenzene. So this is basically the reagent that is used for sequencing the protein. So DFNB, dinitrofluorobenzene, or the correct nomenclature is 1-fluoro-2,4-dinitrobenzene. So you can see a fluorine group is attached in the first carbon here. Then we have at the second and the fourth position 2 nitro groups are attached to the benzene. So that's why 1-fluoro-2,4 dinitrobenzene. Okay. So what it does, basically when we have a peptide, it can actually DNFB can actually react with the terminal amino group, free amino group of the peptide. Okay, so each peptide will have a N terminal and a C terminal, right? So you know how a amino acid is, right? So you have a carbon to which a amino group is attached, a carboxylic group is attached, one hydrogen is attached, and one side chain is there. So when the peptide chain is formed, when the proteins are formed, basically one amino acid is attached to another amino acid by a peptide bond. So you will have CONH and then again the carbon of the second amino acid, again CONH, and so on. So this is how you will have a peptide bond. So this will be the peptide bond. Okay. So now each peptide will have an N terminal and a C terminal. So this DNFB can actually react to the N terminal, and it can help us identify the N terminal amino acid. So if you see here, we have the N terminal, the amino terminal, the first amino acid marked in red. So this 1-fluoro-2,4-dinitrobenzene will react to this amino terminal, and then we can cleave off this part, and we can actually identify this compound that we are getting on either TLC or HPLC and compare it with standard amino acids. So what we can do, we can have all 20 amino acid free amino acids react with DNFB. We can make a standard, we can either separate it on HPLC or TLC. So each amino acid will have a characteristic R group, and thus each amino acid will have a characteristic end product, that is the complex with the DNFB. So based on each amino acid, each amino acid will get separated on TLC or HPLC in its own unique position. So based on that standard information, we can actually analyze what is the first N-terminal amino acid of a peptide. Once this is analyzed, we'll get the free peptide from the second position here. So let's say we have a 5 amino acid peptide, penta peptide. Okay, so we have the first amino acid, second amino acid, third amino acid, fourth amino acid, and the fifth amino acid. Okay, so this is the N terminal and this is the C terminus. Okay, so dinitrofluorobenzene will react to this first amino acid, and then we'll cleave this peptide. So we'll have now 2, 3, 4, 5. Okay, so now this will become the new N terminus, and this product will get separated along with the DNFB. Okay, DNFB will take the first amino acid and we'll get the rest of the peptide here. Next, again, if we add DNFB, what will happen? It will react to the second amino acid, and then we can cleave it off and we can determine what is the second amino acid. So from here we got the first amino acid, then again DNFB will be added, so we will get the second amino acid. Once the second amino acid is removed, we have 3, 4 and 5. So now this becomes the N terminal. Again DNFB will be added, so we will get the third amino acid, then the fourth amino acid and fifth amino acid. Similarly, we can get all 5 amino acid sequence in a peptide. So this is how we do protein sequencing. Okay, for DNA sequencing, we were analyzing each nucleotide by either chain termination or by chemical modification. In this case, this is also type of chemical modification, but this is terminal modification. Terminal amino acid is modified. Okay. Moving on to the next question. What is the result of Edwin degradation method in protein sequencing? Option A, it breaks peptide bonds to release individual amino acids. Option B, it determines the secondary structure of the protein. Option C, it cleaves the disulfide bond in the protein. And option D, it analyzes the protein expression levels. What is the result of the Edwin degradation method in protein sequencing? It breaks the peptide bonds to release individual amino acids. It determines the secondary structure of the protein. It cleaves the disulfide bonds in the protein, or it analyzes the protein expression level. So what do we have? So the correct answer is option A. It breaks the peptide bonds to release individual amino acids. So Edmund degradation is very similar to the Sanger degradation method, where in the Sanger sequencing, what we were using dinitrofluorobenzene, right? Similarly, in the Edmund degradation, just the chemicals change. This also reacts to the N-terminal amino acid. So the principle remains the same, just that in Edmund degradation we are now using phenyl isothiocyanate. So this is phenyl isothiocyanate. Now, like DNFB was reacting with the N-terminus of the peptide, similarly now phenyl isothiocyanate will react with the N-terminus of the peptide, form a cyclic compound. So phenyl isothiocyanate reacts with the terminal amino group to form a cyclic phenyl carbamyl derivative. Okay, that derivative under acidic condition, we can the terminal amino acid is cleaved from the main chain as the thiazolinone derivative. Okay, and that thiazolinone derivative is extracted into the organic solvent and it forms phenyl thiohydantoin amino acid. So we'll have the amino acid R group here, and this R group will differ for all 20 amino acids, and that's how we'll have different phenyl thiohydantoin derivative for each amino acid. And the same way we were doing in Sanger sequencing, we can have a standard here for all 20 amino acids, and based on that we can actually determine what is the first amino acid of this peptide. Then once this amino acid is cleaved off, again this peptide can be used for next cycle of reaction with the phenyl isothiocyanate, and the second amino acid can be determined, then the third, then the fourth, and similarly the whole peptide can be sequenced using phenyl isothiocyanate by making the phenyl thiohydantoin derivative for each amino acid. Okay. So this R group basically is the side chain of the amino acid. So if you see here, so this is the side chain of the amino acid. So if it is a alanine, we will have a CH3 group. If it is a glycine, we will have a H group, and so on. So this R group will basically, now you can see this phenyl isothiocyanate first reacts with the amino group of the peptide, and then the cyclization happen, and once the cyclization happen and it gets cleaved off from the peptide, we will have here the cyclized product with the R group, and this once heated we get the phenyl thiohydantoin derivative of that particular amino acid that was there in the peptide. Okay, and you can see the rest of the peptide is now free. So we have a new N-terminal amino acid. Now that new N-terminal amino acid can again react with this reagent and we can get one more amino acid information. Okay. Moving on to the next question now. Question number 8. What role does a carboxypeptidase enzyme play in a protein sequencing? What role does a carboxypeptidase enzyme play in protein sequencing? Option A, it cleaves the N terminus of a protein. Option B, it cleaves the C terminus of a protein. Option C, it identifies post-translational modification. And option D, it cleaves modified amino acids. What role does a carboxypeptidase enzyme play in protein sequencing? It cleaves the N terminus of the protein, it cleaves the C terminus of the protein, it identifies post-translational modification, or it cleaves modified amino acids. The correct answer is it cleaves the C terminus of a protein. So, till now, all the protein sequencing techniques that you saw for either be with the Sanger reagent, that is the dinitrofluorobenzene, or either the Edmond degradation, that was phenyl isothiocyanate. Both of them were actually recognizing the N terminus of the protein, right? Both of them were reacting with N terminal amino acid and then cleaving the N terminal amino acid off. But what if I want to determine the C terminus of the protein? What can I do? I can use this enzyme carboxypeptidase. So what will it do? Let's say I have a peptide sequence with the C terminus amino acid. So this is the amino acid with the C terminus. So what will happen? The carboxypeptidase will cleave this off, and this free amino acid can then be used for either DNFB based detection or Edmond degradation, and we can determine the amino acid at the C terminus. So basically, if I have a 5 pep, sorry, 1, 2, 3, 4, and then 5. Okay, so this is the C terminus, this is the N terminus. So once I react this peptide with carboxypeptidase, this part will get cleaved off. So I'll get 1, 2, 3 and 4, and I'll get the free 5th amino acid with its own C and N group. Okay, so this free amino group, now this free amino group can react with both DNFB and phenyl isothiocyanate, that is the Edmond degradation. So I can use this 5th amino acid for any of these methods, and I can determine what is this 5th amino acid, what is this C terminal amino acid. Again, remaining peptide can be cleaved off with carboxypeptidase and the 4th. So basically what we were doing in the previous cases, what we were doing, we were sequencing it from this direction. In this case, we can use this enzyme to sequence the protein from this direction. So it is basically this enzyme will cleave at the C terminus of the protein. Okay. Now moving on to the next question. Which of the following is a common method for detecting RNA hybridization in northern blotting? Which of the following is a common method for detecting RNA hybridization in northern blotting? Option A, autoradiography. Option B, fluorescence microscopy. Option C, colorimetric assays. And option D, mass spectrometry. Which of the following is a common method for detecting RNA hybridization in northern blotting? Autoradiography, fluorescence microscopy, colorimetric assays, and mass spectrometry. The correct answer is autoradiography. So this is a common technique that is used to detect the RNA hybridization. So northern blotting is the technique where we detect different mRNA sequences. Okay, so if you remember from the previous sessions, we studied about a technique called Western blotting, where we had the protein sequence separated on the SDS page separated by SDS page, and then the protein bands were blotted on a membrane, and that membrane was uh, then tagged with different antibodies to detect the particular protein sequence, like the particular protein of interest. So that technique was used to check the expression level of the protein, whether the protein is expressed or not, and how much the protein is getting expressed, and so on. So similar thing can be done for mRNAs, whether the mRNA is being expressed or not, what is the expression level of the mRNA. To check that, we can isolate the mRNA, separate it on agarose gel electrophoresis, a denaturating agarose gel, and then different bands will be there on the agarose gel. Those bands of mRNA can be blotted onto a membrane, and that membrane can then be combined with different probes that can detect a particular mRNA that we want to study. Let's say I want to check a particular mRNA expression, so I can use a probe for detecting that particular mRNA, and then we can see whether that particular mRNA is present or not. So that is a technique. So in that case, detection method we generally use autoradiography. For Western blotting, if you remember, the detection was mainly enzymatic color based detection. So we were using secondary antibody that was reacting with that was tagged with some enzyme, and it gave a color at the end, so we can see the blot on the membrane. So how do we radio label the probes actually? So the probes are basically short DNA sequence complementary to the mRNA. So if I have mixture of mRNA and separate it on gel, I'll get different bands based on the size of different mRNA, right? Now let's say my mRNA of interest, like I have certain gene and I want to see I want to see the expression level of that gene, or expression level of the protein in the cell. So basically DNA is transcribed into mRNA, and mRNA is translated into protein inside a cell, right? So if I want to see the expression level of a particular gene, I will see how much mRNA is being produced inside the cell. So for that, I'll extract all the mRNA, total mRNA, I'll separate it based on size, and then I'll use a complementary DNA sequence to this mRNA. So this mRNA will have a sequence, I'll use a complementary DNA sequence. Okay, so that is called a cDNA. Sorry. So we'll use a complementary DNA sequence as a probe. So let's say this is our mRNA. So I'll synthesize a complementary DNA sequence of a certain length that will specifically bind to this mRNA, and this DNA sequence, this will be synthesized, and this will be used as a probe. So before using it as a probe, I have to add some radio label, so that once it binds to my mRNA of interest, I can detect it on the membrane by doing autoradiography. So I'll use radio labels here. So what I'll do, so this is the DNA that we have synthesized, that was complementary to the mRNA of interest that we want to study. We first we first treat it with uh, we first treat it with an exonuclease to create overhangs. Okay, overhangs like this, and then at these overhangs we will add radioactive DATP. So we will add terminal transferase that will transfer this radioactive DATP. So it's a adenine triphosphate, and the one of the phosphate will be radio labeled P32, and once we transfer this DATP, so the end will now have a radioactive group. Okay, this can be easily separated by doing a gel filtration chromatography. So if you remember from the chromatography session, we can use gel filtration to separate anything based on its size. So we can elute this. So this will obviously be larger in size than the unlabeled probe, and also it will be larger in size than the all the reagents that are used. So basically we'll use this sequence, we'll use this to identify the mRNA sequence on the nitrocellulose membrane. So this is the membrane that we'll have. On the membrane all these bands will be blotted, and now I want to see where my probe is binding. So let's say my probe goes and binds here. Then by doing autoradiography, I can see, so this band will be seen in the radiograph as a black band, and none of the bands will be visible. That's how we'll know that okay, this band is present. That means this particular mRNA is present in the sample. This particular gene is being expressed. So that is a basic idea of how northern blotting is done and how radio labeling of a probe is done. Okay. Now moving on to the next question, question number 10. Dash affinity column is used for purification of mRNA. Dash affinity columns are used for purification of mRNA. Option A, poly A. Option B, nickel NTA. Option C, poly T. Option D, dextran. Dash affinity column is used for purification of mRNA. Option A, poly A. Option B, nickel NTA. Option C, poly T, and option D, dextran. The correct answer for this question is option A, poly A. Sorry, option C, poly T. I'm sorry. So this, why is this used for purifying mRNA? So if you know the basic structure of mRNA, so mRNA has a 5' cap, and then 5' UTR region, then the coding sequence, then 3' UTR region, and then at the 3' it has a poly A tail. This poly A tail is basically long chain of multiple A residues added after the 3' end. So these are basically modifications done on the mRNA in order for its proper translation process. So this poly A tail can be of varying length. So it can be of 200 residues, it can be of 100 residues. So it can have variable lengths, and this is the feature that we use to actually isolate the mRNA from the sample. So once we have the cells and we want to extract the mRNA, we will lyse the cells, take out the RNA content, total RNA content, and then we'll pass it through a poly T column. So poly T column will be basically beads connected with multiple T residues, okay, thymine residues. So you know adenine is complementary to thymine. So adenine forms hydrogen bonds with thymine and guanine forms hydrogen bonds with cytosine, right? So if there is a resin bead that is connected to multiple T residues, so wherever it will find adenine residues, it can easily make complementary bonds. It can have complementarity with that poly A chain, and the mRNA can be bound to this resin. So mRNA will bind to this resin, rest all the other types of RNA will get eluted, and later we can just break these hydrogen bonds to release the mRNA from the beads, and that's how we can get purified mRNA sample using the poly A chain that is naturally present at the 3' end of the mRNAs. Okay, so it has a 5' cap structure, coding sequence, and a C-terminal poly A tail. This nucleotide A forms 2 hydrogen bonds with nucleotide T, and this pairing is very specific. Exploiting this feature, mRNA population can be isolated from the RNA pool using the poly T affinity column. So that affinity column basically has a resin bead having a poly T tail. Okay, so multiple T residues are attached like a tail, and this T residue will like this T residue will bind to this poly A tail of the mRNA that is present, and then later we can break the hydrogen bonds to elute the mRNA, purified mRNA from the affinity column. So this is a type of affinity chromatography. So if you remember the sessions from the chromatography topic, you will understand how this is done. If you don't remember the affinity chromatography session, I would request all of you to go back and listen to those lectures so that you understand how this purification of mRNA is done. Okay, moving on to the next question now. Question number 11. What is the significance of pre-wetting the nitrocellulose membrane before mRNA transfer in northern blotting? What is the significance of pre-wetting the nitrocellulose membrane before mRNA transfer in northern blotting? Option A, it prevents RNA degradation. Option B, it reduces background noise and hybridization. Option C, it increases mRNA electrophoretic mobility. And option D, it enhances mRNA binding to the membrane. What is the significance of pre-wetting the nitrocellulose membrane before mRNA transfer in the northern blot? Prevents RNA degradation, it reduces background noise and hybridization, it increases mRNA electrophoretic mobility, or it enhances mRNA binding to the membrane. Correct answer is option D. It enhances the mRNA binding to the membrane. So as I mentioned earlier, in northern blotting we basically separate the mRNA on the gel and then transfer the mRNA bands on the nitrocellulose membrane. So this is how we transfer it. So we have the agarose gel on which the bands have been separated. We'll keep the nylon membrane, nitrocellulose membrane over the agarose gel, and we'll stack a bunch of filter paper over it, and we'll keep a weight, and we'll keep this whole thing in a transfer buffer. So what will happen, this buffer will rise slowly due to capillary action. This buffer will rise slowly, and along with that the mRNA will dissolve in the buffer and move to the nylon membrane first. Since all these filter papers will soak the transfer buffer upwards, so there will be a capillary action. So the buffer will rise upwards, and along with the buffer the mRNA bands will also rise upward. But what will happen, the mRNA band will get stuck in these nitrocellulose membrane, in this nylon membrane, and the buffer will only be able to pass through the nitrocellulose membrane. So that's how from the agarose gel the mRNA bands will be transferred on the nitrocellulose membrane, and the buffer will go to the filter paper. Okay, so this is how the transfer is done. And before doing the transfer, we generally pre-wet this nylon membrane so that it enhances the binding efficiency of the mRNA to the membrane. I hope that's clear. Moving to the next question, question number 12. In northern blotting, the autoradiogram is developed at option A, room temperature, option B, 40 degrees Celsius, option C, 80 degrees Celsius, minus 80 degrees Celsius, and option D, 80 degrees Celsius. In northern blotting, the autoradiogram is developed at option A, room temperature, option B, 40 degrees Celsius, option C, minus 80 degrees Celsius, or option D, 80 degrees Celsius. So the correct answer for this question is option C, minus 80 degrees Celsius. We have to do this development at a very low temperature so that we get a very specific signal. So basically in autoradiography we are visualizing the membrane under X-ray radiation, right? So in that case, if we do it at room temperature, we can actually see a very high signal, and that can actually give us false results, because what will happen if, let's say, this is our membrane, okay, and we have a band here that is radioactive and that is showing the signal. Now if we do the development of the autoradiogram at a low temperature, we'll see signal something like this. But if we do it at a room temperature, we'll see a signal something like this, because there will be a lot of beta emission, so the whole part will be showing radioactivity instead of a single band. So if we see a big blotch like this, we can't determine if this is this fragment or this fragment or this fragment. Okay, so let's say in the agarose gel there were 3 fragments, and now after the development only 1 fragment is actually radioactive. That means this is the mRNA that is actually the mRNA that we are interested in studying. But if we see a big blotch like that is covering all 3 bands, we won't be able to determine which is the mRNA that is actually getting expressed inside the cell. So that's why the development is done at a very low temperature, so that we can get very precise signal for the radioactivity. Okay. Moving on to the next question now, question number 13. After RNA transfer in northern blotting, what is typically done to fix the RNA on the membrane before probe hybridization? After RNA transfer in northern blotting, what is typically done to fix the RNA on the membrane before probe hybridization? Option A, UV cross-linking. Option B, ethanol precipitation. Option C, heat denaturation. And option D, alkaline treatment. What is generally done to fix the RNA on the membrane before probe hybridization? The correct answer is option A, UV cross-linking. So as I have already mentioned, in the northern blotting, we are transferring the RNA bands on the nylon membrane, and then we add the probe, that is radio labeled probe, and the probe gets hybridized to wherever the complementary mRNA sequence will be there, and then we visualize it by autoradiography, right? So in that technique, after the transfer and before hybridization, we have to do UV cross-linking of the RNA so that the RNA is completely fixed in the membrane, because when we are doing this probe hybridization and then autoradiography, the conditions are harsh and we don't want to lose the mRNA. So if the mRNA goes out of the membrane under certain conditions, we will not be seeing a positive result, and we that can actually lead us to wrong conclusions. So let's say the mRNA of interest was there and it somehow got out of the membrane, so that will show us that the mRNA is not there, but the mRNA was there in the sample. So that's why it's better to fix the RNA on the membrane before doing the probe hybridization, so that it doesn't escape the membrane. So that is done by UV cross-linking. Okay, so this is what I was talking about. So the sample, this is the mRNA sample. So this will be separated on the agarose gel electrophoresis. So we generally use denatured agarose gel for running mRNA sample, if you remember from the electrophoresis classes also. So we use denatured agarose to denature the mRNA secondary structure, so that it can actually get separated based on the size. If the secondary structures of mRNA are present, they will run to the very end of the agarose gel, and we will not be able to see separate bands for each mRNA. Okay. So after the separation, the bands are transferred on the nylon membrane, as I have explained previously. Then this is used for hybridization. So between this step, we do UV cross-linking. That is basically done to fix the RNA onto the membrane. Okay, fix the RNA on the membrane. We do UV cross-linking. So after that, we then add the radio-labeled probe. So I have explained how we add the radio labels to the probe. This probe is added onto the membrane. So wherever the complementary sequence will be there, the probe will bind to those sequences, and then we will analyze using X-ray, because it's a radio labeled probe. So we'll use X-rays to visualize it, and the bands where the probe has hybridized, where the probe has bound, they will be visible because they will be radioactive, so they will be visible under X-ray. Okay, so this is a simple schematic for northern blotting. I hope that's clear. Moving on to the next question, question number 14. What is the primary purpose of southern blotting in molecular biology? Option A, to analyze protein-protein interactions. Option B, to determine the sequence of DNA fragment. Option C, to detect and analyze specific DNA sequence in the sample. Option D, to quantify RNA expression levels. What is the primary purpose of the southern blotting in molecular biology? Option A, to analyze protein-protein interaction. Option B, to determine the sequence of the DNA fragment. Option C, to detect and analyze specific DNA sequence in a sample. And option D, to quantify RNA expression levels. The correct answer is option C, to detect and analyze specific DNA sequences in a sample. So like northern blotting was done for RNA, southern blotting is generally done for DNA. So in northern blotting, we were actually checking the mRNA expression levels. In southern blotting, we can actually analyze a specific DNA sequence. So if we have the whole genome and I want to see if a particular gene is present in that genome or not, I can actually use specific probes for that particular gene, and I can fragment the whole genome into certain pieces, run it on agarose gel electrophoresis, and then use the probe for particular gene, and then see if that probe binds to the gene. We will see a signal on the autoradiograph. So basically to detect and analyze specific DNA sequence, we do southern blotting. So it is very similar to northern blotting. Instead of RNA, we have DNA sample here. So this is the schematic for southern blotting. So you can see, first is genomic DNA isolation. Obviously, if we want to check a particular gene is present or not, we need to isolate the genomic DNA first. After isolating the genomic DNA, we digest it to generate certain fragments. So we'll do the digestion so that we'll have fragments throughout the gel. We don't want fragments on the top and bottom part of the gel without having any fragments in the middle. So we want the fragments to be evenly distributed throughout the gels. Then we'll separate these fragments on the agarose gel by electrophoresis, and then we'll transfer these bands onto the nitrocellulose membrane. So same process how we were transferring the mRNA bands, we can transfer the DNA bands also from the agarose gel. Once the bands have transferred to the nitrocellulose membrane, we'll hybridize with radio labeled probes. So same how we were radio labeling the probes in case of mRNA, in case of DNA also we can have complementary DNA sequences, small probes that will have the radio label, and we can use that to do the hybridization. Wherever the probe will bind, we can visualize under in autoradiogram under X-rays to see whether that particular DNA is present or not, that particular gene is present or not in the genomic DNA. So that is a basic schematic for southern blotting. Okay, I hope this is clear to everyone. I'll move on to the next question now. Question number 15. Which of the following is an advantage of vacuum transfer over capillary transfer in southern blotting? Which of the following is an advantage of vacuum transfer over capillary transfer in southern blotting? Option A, faster transfer times. Option B, lower cost of equipment. Option C, better control over transfer conditions. And option D, less risk of sample contamination. Which of the following is an advantage of vacuum transfer over capillary transfer in southern blotting? Faster transfer times, lower cost of equipment, better control over transfer condition, or less risk of sample contamination. So correct answer for this is faster transfer times. So till now the transfer mechanism that I have explained for northern blotting and southern blotting is based on capillary transfer. So basically this is the capillary transfer mode. So we have a support block, we have a filter paper that acts as a wick. So in this tray we have the transfer buffer, and this filter paper is actually touching the transfer buffer. So by capillary action, the buffer will rise up on this filter paper, and over this filter paper we have kept the gel here. So the blue colored thin slice you can see. So this is the gel. Then over that gel we'll keep the nitrocellulose membrane, and we'll stack it with a bunch of paper towels or filter papers. So what will happen, this buffer will rise through this filter paper that is acting as a wick. Buffer will rise through this filter paper and reach the gel. The DNA bands will get dissolved in the buffer and move up, because these filter paper, the stack of towel or paper towel or filter paper, whatever is there, they will also have the capillary action, so they will also pull the buffer up. So along with the buffer, the DNA bands will also move up. But since we have kept a nitrocellulose membrane, the DNA bands will get stuck on the nitrocellulose membrane, and only the buffer can move up to this stack of towel, and the DNA bands will get stuck in the membrane. So that's how we transfer the DNA from the agarose gel to the membrane. And this capillary action can go on for 16 to 24 hours, so that all the DNA from the gel is transferred to the nitrocellulose membrane. Okay, so once the whole stack of towel is wet, we can actually replace it with a drier stack of towel and then again continue the capillary action. So it's a capillary action, so you can imagine it's a very slow process, right? As opposed to that, we have another technique called vacuum transfer, which is basically the same thing, but now instead of depending on the capillary action of the buffer rising through the paper, we have vacuum attached to it. So we have the gel, we have the membrane, then the blotting paper, and we have vacuum. So vacuum will pull down the buffer quickly. So the whole process is now sped up basically. So basically we are using vacuum to pull the buffer, and along with the buffer the DNA will move from the gel to the membrane. So that will make the whole process faster. That's why vacuum transfer is more efficient, because this process can be completed in I think 2 hours or so, whereas in capillary transfer we have to wait for up to 24 hours for the whole transfer to happen. Okay, so it's a very lengthy process if we are going by capillary transfer, whereas if we are doing vacuum transfer, it's a very it's a quicker process. Okay, so that's why vacuum transfer is more efficient than the capillary transfer. Moving on to the next question now. What are the basic base specific cleavage sites used in Maxim-Gilbert method? So we have discussed the Maxim-Gilbert sequencing method. So what are the basic base specific cleavage sites in the Maxim-Gilbert method? So if you remember, in the Maxim-Gilbert method we were doing chemical modification of the bases. So there were 4 types of chemical reactions that were happening, that was specifically modifying particular nucleotides. So what were those 4 base specific cleavage sites? The correct answer here is option D. So we had a reaction that was specifically modifying guanine. We had another reaction that was specifically modifying the purine, so both adenine and guanine. Then we had another reaction that was specifically modifying the cytosine residues, and the 4th reaction was modifying both cytosine and thymine. So if you remember, then we were separating the different fragments on the gel electrophoresis and based on the sequence. So if the band is there in both adenine plus guanine reaction and in the guanine reaction, we will consider it for G. If the band is only there for adenine plus guanine but not in the guanine, then we will consider it for adenine, that is A. Same for T and T plus C and C. Okay, so I have already explained this, so I hope this is clear to everyone. So we have 4 types of reaction: A plus G, C, G and T plus C. Okay, so moving on to the next question now, question number 17. Starting from the sequencing primer, what is the sequence of the DNA sample? So this is a typical Sanger sequencing gel picture. So we have to determine the sequence of the DNA based on the bands that you can see. So if you remember, in the Sanger sequencing, it was based on the chain termination method, right? So we were adding the DD-NTPs, so DD-GTP, DD-ATP, DD-CTP and DD-TTP. So all these 4 dideoxynucleotides were added separately in 4 different reactions, and those 4 reactions are then separated on the agarose gel electrophoresis, and we can actually determine the sequence based on where it is where the sequence is terminated. Okay, where the DNA synthesis is terminated. So if you see here, the smallest fragment is coming from the G residue. So smallest fragment is coming from the G residue here. The second is this one, that is T. So the next will be T. Third residue here is A. So this is how we have to read. We have to start reading from the bottom, because whatever the sequence will be, so if the sequence is C, A, T like this, so the complementary will be G, T, A like this, right? So whenever the incorporation will happen, so let's say dideoxy GTP is added, so that means the chain termination will happen here, and we will get the smallest fragment, that is single nucleotide G. Then once dideoxy TTP is added, the chain termination will happen here, and this fragment is basically GT. So this is G, this is GT, then this fragment is GTA. Then next is C. So we have C, again C, and again C. So 3 times C. So this fragment is basically GTAC. Then the next fragment is GTACC. Then this is GTACCC. Okay. Then after that we have again a G. So this fragment will be GTACCCG. Okay. Then we have 3 A's consecutively. So A, and A. So this fragment is basically GTA CCCGA. Okay, this fragment is GTA CCCGAA, and this fragment is GTA CCCGAAA. Okay, then the next residue is T. So we'll have T. So this fragment is basically corresponding to G, T, A, triple C, G, triple A, then T. Okay. Then next is C. So that means this is G, T, A, triple C, G, triple A, then T, then C. Okay. So similarly this is A. This will be GTA triple C, G, triple A, T, C, A. Okay, then we have 2 G's, then we have A. So the sequence is GTA triple C, G, triple A, T, C, A, G, G, A. So this is how you have to read. You have to start reading from the bottom and see what is the smallest fragment, then what is the second in number, third in number, and so on. So which is the correct answer here? Here the first option is the correct answer, option A. GTA triple C, G, triple A, T, C, A, double G, A. Okay. So I hope this is clear how to read a sequence from the gel. So if you are doing this experiment on your own, you might have to analyze your gel. So this is how you will proceed. Moving on to the next question, question number 18. In the experiment, DNA is finally recovered by precipitation of the supernatant width. So when we are doing DNA extraction, so I have told you in southern blotting, we need to do whole genome extraction first, right? So when we are doing DNA extraction, how do we finally precipitate the DNA from the supernatant? So final extraction, the DNA is in supernatant. Now we have to precipitate the DNA so that we can resuspend it in water at a very concentrated level. So how do we precipitate it? Option A, methanol, option B, ethanol, option C, acetic acid, and option D, acetone. What do we use out of all these 4 options to precipitate the DNA from the final supernatant while doing DNA extraction? So the correct answer is option B, ethanol. So ethanol is used for the final precipitation. So this is how we do the whole genome uh, DNA extraction. So we take the cells, lyse the cells, break down the cells, basically do different enzymatic digestion, precipitate the DNA with absolute alcohol, then purify it with chloroform, phenol chloroform extraction. So phenol chloroform will basically separate the different protein and lipid residues from the DNA. So we'll get the purified DNA in the aqueous phase, and then from from that we need to precipitate the DNA again. So we'll use absolute ethanol, absolute alcohol, or ethanol to precipitate the DNA, and then we can resuspend that precipitated DNA pellet in desired concentration, desired amount of water to get a desired concentration. And this is how a genomic DNA looks like on agarose gel. So genomic DNA is a very large fragment of DNA, so you can see it is running very close to the well. It doesn't reach to the bottom of the gel. It somehow remains at the near the well only, because it's a very large fragment of DNA. Okay. Moving on to the next question. Question number 19. The deep urination treatment in blotting involves the use of HCl and option A alkali, option B acid, option C proteins, and option D nucleic acid. Deep urination treatment in blotting involves the use of HCl and option A alkali, option B acid, option C proteins, and option D nucleic acid. So the correct answer is alkali. So deep urination treatment is done in blotting to allow transfer of wide range of DNA fragment sizes. So basically during southern blotting, so if the DNA fragment is very large in size, to allow complete transfer of all the DNA fragments, whether large or small, to the nitrocellulose membrane, we first treat the gel with deep urination deep urination buffers. So we treat the gel with HCl followed by alkali to neutralize it, and then we start the transfer onto the nitrocellulose membrane. Okay. Moving on to the next question. This is the last question of this session. Question number 20. Which of the following techniques would be most useful to study gene expression? Option A southern blotting, option B northern blotting, option C western blotting, and option D eastern blotting. Which of the following technique would be most useful to study gene expression? Okay, so the correct answer is northern blotting. So you have to pay attention. It is not, we don't want to study gene, we want to see the gene expression. That means we want to study the mRNA levels. Okay, so if a gene is getting expressed, that means it is getting transcribed into mRNA, and then mRNA is translated into protein, right? So we want to study the amount of mRNA. So amount of mRNA is high, that means the gene expression is high. Amount of mRNA is low, that means the gene expression is low. Okay, southern blotting is done to identify a particular gene is present or not in the genomic DNA. Okay, western blotting is done to identify a particular protein is present or not in the particular sample. And eastern blotting is actually done for identifying carbohydrates and lipid molecules. So lipoproteins or glycoproteins are generally analyzed by using eastern blotting. So northern blotting is the technique that is that would be most useful to study gene expression. Okay, so with this we come to the end of the session. So I hope I have covered all the topics that are related to the week 11 assignment. If you guys have any doubt, you can ask me anytime. So I'll post the recording of this session on YouTube, and you will get the link for that, and I'll share the slides with you through the Drive link. So I hope the session was useful to all of you. So with this I'll end this session now. Thank you so much.